The protein folding problem has plagued scientists for years. Protein folding is essentially how an amino acid sequence within the DNA leads to a tertiary structure, or three-dimensional shape. What makes accurate protein folding so challenging is that the 20 different amino acids that build proteins interact with each other in very complex ways. Adding to this challenge is the fact that proteins often have hundreds or thousands of amino acids. Taking all these factors into account, mapping out a correct protein shape is almost impossible for any one biologist or team of biologists to do accurately and within a reasonable amount of time. That is, until recently. In this video, we are going to discuss recent changes in protein folding technology, specifically to do with machine learning, and how this changes the game, not only for the mapping of proteins, but also for biology and biotech in general. First off, we want to go a bit more in-depth into what exactly protein folding is. Protein folding is the way that a protein goes into its lowest energy, tertiary 3D state. The protein instantly goes into the shape after being completely processed. It is then ready for use in whatever cell or part of the body it is needed in. What governs how a protein folds is its amino acid sequence. Every amino acid contains the same amino acid backbone, that is, an amino acid group on the left that connects to carbon in the center that connects to a carboxylic acid group to the right. But each amino acid has a different R group. Each R group has a different chemical composition which leads to different intermolecular forces present in each amino acid. For example, cysteine has a sulfur atom at the end of its R group, which allows it to make a disulfide bridge to another cysteine. Amino acids with hydrogens, like hydrogen bonding with oxygens on other amino acids. Electrically charged amino acids can also make ionic bonds with amino acids of the opposite charge. There are many other intermolecular forces that are present in some amino acids, further complicating protein folding prediction. Amino acid sequences are derived from triplet codes on a DNA strand. One amino acid per 3 nucleotides. Example given would be TGT and TGC coding for cysteine. So the way that amino acid sequence is made determines what amino acids are close to each other and which aren't. Basically, the amino acids have to be in that order. Because amino acids are sequenced in a specific way, theoretically, you could predict what a protein looks like just by looking at the amino acid sequence associated with said protein. The glaring issue with this method is that many proteins have extremely long sequences, as well as the fact that there are 20 different amino acids, each with different intermolecular forces that impact the protein's shape. This makes it almost impossible for scientists to predict the shape, and we can only really know what the shape is by studying the protein with methods such as X-ray crystallography or NMR spectroscopy. Understanding how proteins fold is important because the shape of a molecule is essential to understanding how it works. Let's use an example from a very simple molecule, methamphetamine. As many of you know, methamphetamine is a potent drug. However, methamphetamine's structural isomer is levomethamphetamine, a commonly used nasal spray. The only difference between these two molecules is that one is flipped horizontally and the other vertically. This ever so slight structural change emphasizes the importance of understanding a protein's 3D shape, as it can have such a large effect on its function. Even a temperature change is enough to alter a protein's 3-dimensional shape, completely destroying its function. Put simply, the more we can understand the 3-dimensional shapes of proteins, the more control we have over our bodies and what happens inside of them. Now that we have established the basics of protein folding, as well as its crucial importance, we can look at the modern methods used to address this difficult problem. Perhaps the most famous event surrounding protein folding is the Casp Competitions. The Casp Competitions, which stand for Critical Assessment of Protein Structure Prediction, is a competition that takes place every 2 years. According to their website, Casp experiments aim at establishing the current state of the art in protein structure predictions, identifying what progress has been made, and highlighting where future effort may be most productively focused. The competition is open to the whole world and seeks to move the field of protein folding technology forward. Many scientists view the competition as the world championship in this specific field of science. Because of this, the results of Casp are a reliable indicator of how protein technology is evolving. For the vast majority of the competition's history, the accuracy of protein folding has been very unreliable. That is, until recently. For the last 2 years, one competitor has risen far above them all, and has exponentially heightened the overall accuracy of the protein folding predictions in the competition. This competitor is AlphaFold, a subdivision of the AI firm DeepMind, which is owned by Google. This is not the first time DeepMind has made headlines with its advanced intelligence technology. AlphaGo, another creation by the firm, became the first computer program to defeat the world champion of Go, a game even more complicated than chess. DeepMind has also made AlphaFold software available for users across the world, releasing a very powerful tool with unforeseen potential in the future of medicine and beyond. That is, if your computer can run the software. AlphaFold uses a type of AI software called machine learning. Machine learning is a process computers use to complete tasks without having to have the tasks be explicitly programmed by someone. The computer finds patterns within the data it is given and uses these patterns to make and execute predictions. Because the success of machine learning requires data, the more data the program has, the better. This is why machine learning, specifically from Google's DeepMind, is better than the best humans at chess. Initially, it was not, but the more simulation games the computer would go through, the more data it would gather about said game, and then learn from that. No human can take as much data and consistently find and use patterns as a machine learning program. AlphaFold's machine learning program works on the data of protein structure in 3D shapes scientists get from their X-ray crystallography models. These images and models are fed into the software, along with the amino acid sequence of said protein, and the AI takes in that data and learns from it to see patterns in how amino acid sequences lead to a three-dimensional protein shape. AlphaFold's ability to predict the three-dimensional structure with increasing accuracy is a massive moment for the world of biology and computer science. AlphaFold may prove invaluable in many sectors, such as pharmaceuticals, where it can help with the creation of new drugs. Many of the uses for this tool, however, are hard to predict. But with more control and understanding of something as essential as proteins, there seems to be near endless possibilities.